Hello, I'm Dr. Aizaz from MedicoVisual.com, and in this visual lecture we will talk about gastrulation. Here is the embryo at the start of third week of development. If we cut open this embryo, if we divide the embryo with a knife into two parts, and if we remove this part of the embryo and we observe this cross section of the embryo, what we will see is that it consists of two types of cell forming the bilaminar germ disc. The upper layer of cell is called epiblast, while the lower layer of cells is called hypoblast. Amniotic cavity surrounds this embryo from above, while the secondary or definitive yolk sac surrounds this embryo from below. Then this yolk sac membrane or yolk sac endoderm, it surrounds this yolk sac. Of course, then there is extra embryonic mesoderm, and whole this structure it is floating freely within this chorionic cavity, but it is still connected with the connecting stalk. And of course, whole this structure is surrounded by cytotrophoblast cells, and of course outside this will be the syncytiotrophoblast, which has not been shown in this diagram. Let's focus on this bilaminar germ disc. If we undo the cut here, we will see that it is a disc-like structure consisting of hypoblast as well as epiblast layers, and this is called bilaminar germ disc. Let's focus on this. So here is this bilaminar germ disc, and let's make a copy of this bilaminar germ disc, and in this copy version let's cut it from here. So if we cut it from here and we observe it from front, for example, you are standing here and you are observing it from front, how it will look like. So here is this bilaminar germ disc from front. Again, it consists of epiblast layer as well as hypoblast layer. Now at this stage what happens, and of course we are talking about third week of development. So at the start of third week of development, what will happen is that this epiblast layer, they will release some special substances in between these two layers, in between the hypo and epiblast layer. What is this substance? It will release lots of hyaluronic acid in between these two layers. This hyaluronic acid is a hygroscopic compound. It can bind with and it can pick up lots of water molecules, and thus a fluid-filled space will be formed in between hypo and epiblast cell layer. And in this way, we will get a blotted appearance of the embryo. Here we can see that we have this blotted up appearance of the embryo. Then these midline epiblast cells, they will release some chemoattractants, and what these chemoattractants will do is that they will cause growth of epiblast in the periphery. Now what these chemoattractants will do is that they will cause proliferation of epiblast in the periphery, and they will attract these epiblasts, these newly formed epiblasts, towards the midline. So as these newly formed epiblasts aggregate in the midline, what we will see is that in the caudal midline there will be formation of a streak of cells. It will start in the mid of caudal midline and it will extend caudally as well as it will extend cranially. And at the cranial end it will form this swelling or node called primitive node. And this streak of cell, it is called primitive streak. So here is this structure called primitive streak on the cut section, as well as while we view it from the top, and this node is called primitive node or Hensen's node. Remember that this structure is formed only at caudal end. Caudal end means tail end. Of course we humans do not have tail, but still traditionally embryologists call it caudal end and this end as cranial end. So with the formation of primitive streak, we can clearly say that this end is caudal end on which the primitive streak is formed, while the other end that is opposite to primitive streak is the cranial end. Before the formation of primitive streak, just by looking at this bilaminar germ disc, we cannot tell that which end is the cranial and which end is the caudal end. Along with that, in the midline as this primitive streak is formed, we can say that this is the right side of the embryo and this is the left side of the embryo. So axis determination is one of the important functions of the primitive streak. Now here an important question arises that why primitive streak only forms at the caudal end? What stops the formation of primitive streak at the cranial end? Actually, we have these hypoblast cells, and these hypoblast cells, they are like wise old men. They give important instructions to the overlying hypoblast cells. The hypoblast cells of the cranial end inhibit the formation of primitive streak at the cranial end of epiblast. As they do so, the epiblast in the caudal end, they quickly forms the primitive streak as they are so keen to do so. This cranial end hypoblast also helps in formation of head structure, so it is called anterior visceral endoderm. Anterior? Where the fuck is interior here? Of course this is the cranial end, this is not the interior end, so why we call it anterior visceral endoderm? I think it should be called cranial visceral endoderm rather than anterior visceral endoderm. Actually, this name is given to this structure by the mouse embryologist, and in mouse, this structure is not just cranial but it is also anterior. Well, let's see how. So here is a mouse and it has to pass through this door. The first structure of his body that will cross the door is its head. So it is called interior. I mean the head of embryo is interior. Interior means something that comes before the other part. And the last part of his body to cross this door is his tail. So his caudal end or his tail is also the posterior end. So in mouse, anterior as well as the cranial, both of these terms are synonymous. Similarly, the term caudal as well as posterior, both of these terms are synonymous. But this does not happen with humans. Because let's say if this human has to cross this door, both the head as well as, of course humans do not have tail, both the head as well as his buttocks or hips, both of them will cross the door at the same time. So we cannot demarcate the head and hips in terms of anterior and posterior. So in humans the head is cranial but it is not necessarily interior. But still conventionally we call it anterior visceral endoderm and you have to do the same, right? Epiblast cells, just like any other type of epithelial cells, they are closely bound with each other, kinda like holding hands of each other, and that is due to special proteins called E-cadherin proteins that keeps them bound together with each other. Now what happens that as these primitive streak cells come into the midline in the center, these midlines epiblast cells, what they do is that they downregulate the E-cadherins. Thus they lose their hands and they are no more bound with each other. So what happens here in this strange land of primitive streak, these epiblasts, these midline epiblast cells, what they do is that first they invite these guests into this area and then they remove their hands. And of course now what will happen that they will not be able to stay together. So let's suppose that this midline epiblast cell, it loses its E-cadherin molecule, so it will tend to fall into this space. And as it does so, it loses its particular shape and it forms a bottle-like shape, a flask-like shape, and now it is called bottle cell or flask cell. So ultimately it will fall into this space and now we call it mesenchymal cell. This is the epithelial cell and this is a mesenchymal cell. The difference between epithelial cell and mesenchymal cells is that number one, the mesenchymal cell, it has an irregular shape, but epithelial cell, it has a particular shape. For example, this epiblast cell, these epiblast cell, they are columnar in shape, but this mesenchymal cell, it has some irregular shape. Secondly, the epithelial cells, they are tightly bound together, they are closely packed together with each other, but that is not true for mesenchymal cells. So what we can say is that as this epiblast, it invaginated inward into this space, what happened with this epiblast cell is that it underwent epithelium to mesenchymal transition. So as more and more cell invaginate inward from this point, here we will see that a small depression is formed here. Here you can see there is a depression. The name of this depression is primitive groove. So more and more cells will come into this space, and here the structure that is formed is mesenchyme. Mesenchyme is basically the embryonic connective tissue. It consists of ground substance that is filled up with hyaluronic acid and other substances, and within this ground substance mesenchymal cells are loosely arranged. On this 3D diagram you can see that this was the primitive streak, and as the cell invaginated inward, a groove is formed in the center of this primitive streak. So this groove is named as primitive groove. Here on the cut section you can see this primitive groove, and on the 3D diagram you can also see the primitive groove. This primitive groove, it extends caudally into another groove, a shallow depression that is called primitive pit. So that there is that primitive pit. Now how this primitive pit forms, what happens that here also some cells invaginate inward, right? Some cells invaginate inward and they move forward in between the space between epi and hypoblast, and in this way this rod-like structure will be formed here. Below this epiblast layer, this rod-like structure is called notochord. We will discuss that how it forms and what is its function in the next lecture, right? And regarding the primitive groove, as you know that how it forms, the cell invaginates here from this groove, the cell invaginates inward, and this leads to formation of this depression called primitive groove, right? So now we have three layers. This is the epiblast layer, this is the hypoblast layer, and in between them there is another layer of mesenchymal cells, and now we have this three layers. Is this the process called gastrulation? Well, no, it is not that simple, unfortunately. What happens here is that as the embryo is growing in size, the simple diffusion process is not simply enough to meet the nutritious need of the embryo. So this is the right time that blood cells should form. As you know that here we have this yolk sac endoderm or yolk sac membrane, and outside this yolk sac endoderm is extraembryonic splanchnopluric mesoderm. Now these mesenchymal cells, they leave this space and they come here into the place of hypoblast, and they displace these hypoblasts into the yolk sac endoderm. So hypoblasts are displaced to yolk sac endoderm, and here these mesenchymal cells come. So these mesenchymal cells will almost completely replace the hypoblast, and those hypoblasts, they will then be displaced to the yolk sac endoderm. These mesenchymal cells, as they come here, they will not remain mesenchymal. They will then again undergo mesenchymal to epithelial transition, and here they will form the definitive endoderm. They will acquire the hypoblast-like shape. Of course, they are not hypoblast. They will acquire hypoblast-like shape and they will form the definitive endoderm. And what about the hypoblasts that were displaced here into the yolk sac endoderm? As I have told you, the hypoblasts, they are like wise old men. They have a real bossy nature. What they will do is that they will order the overlying extra-embryonic mesoderm that, hey, you extra-embryonic mesoderm, start creating blood island. And of course, it will do so. So blood island will be formed in the extra-embryonic splanchnopleuric mesoderm on the order of yolk sac endoderm. In books, what they say is that the underlying yolk sac endoderm induces the formation of blood island into the extraembryonic splanchnopleuric mesoderm. What is the function of blood island? For now, just remember that it helps in formation of blood cells. We will discuss the details of blood island in upcoming lectures. So as these mesenchymal cells that were lying here, they displaced the hypoblast and came here and differentiated into the definitive endodermal cells, this space is now empty. So more epiblast cells will proliferate and invaginate inward. They will migrate towards the midline and invaginate inward through the primitive groove, and again they will come into this space to form the definitive mesoderm. The definitive mesenchyme will be formed here. So now ultimately we have three germ layers due to the faithful work done by the primitive streak. As the primitive streak has performed its function nicely during the 4th week of development, primitive streak usually disappears. But unfortunately, sometimes the primitive streak fails to disappear and the remnants of primitive streak remains and it forms what we call as Sacrococcygeal Teratoma. Here is a newborn with a Sacrococcygeal Teratoma in the hip region here. This is called teratoma. Teratoma means monstrous tumor because it looks like a monster. As you know that these epiblast cells and this primitive streak, it is the precursor of all different types of cells. It forms the three germ layers that are ultimately going to form the complete embryo. So this tumor, it consists of all different varieties of cells. There may be nail, there may be teeth and hair and all those types of adult cells. So it may look like a monster. So that is why we call it teratoma, the monstrous tumor. So now here we have three germ layers. The outer layer is called ectoderm, the inner layer is endoderm, and in between them is the mesoderm. Meso mean in the middle, that comes in the middle, and it consists of mesenchymal tissue. So this is trilaminar germ disk. And the origin of all these germ layer is epiblast cells. This process of formation of trilaminar germ disk is termed as gastrulation. Gastrulation? There is no formation of stomach here. There is no formation of gut tube here. Why the hell we call it gastrulation? Well, this is confusing, so let me explain why it is called gastrulation. Actually, this process was first discovered in small animals, for example, sea urchins. And this primitive organism, their blastula, it consists of a hollow spherical ball of cell. And during the process of gastrulation, their process of gastrulation is very simple. It looks like that someone has pinched here with a thumb, right? So someone has pinched here with a thumb. So here they will invaginate inward, as you can see here that here it has invaginated inward, and in this way what happens is that here this part here we have formation of primitive gut or archenteron. The water and food material, it enters first into this primitive gut and it is released outside through this blastopore. So during gastrulation, what is happening here is that there is formation of primitive gut during the process of gastrulation. But along with that, we have also the formation of germ layers. Here is the endoderm that has formed. Along with that, there is ectoderm. And in some organism, in between them, the mesoderm is also formed. So three germ layers are formed here. Along with the formation of three germ layers, the primitive gut is also formed. So scientists initially thought that the formation of primitive gut, that is gastrulation, is the most important process here. So they simply ignored the dry lamination. So rather than calling this process as dry lamination, they simply call it gastrulation. Why? Because in small organism primitive gut is formed here. But no such process occur in human. In human, during the gastrulation process, no gut forms. But still somehow it is called gastrulation. Why so? By now you must have understood this. Yes, you got it right. Conventions, my friend, conventions, right? So finally, let's review it. So first of all, this is the epiblast layer. What happens that it releases chemoattractants that causes proliferation and migration of epiblast from the periphery into the caudal midline, and then it downregulates the E-cadherin. Thus these epiblasts, they tend to fall into this space, firstly forming the bottle cells, and then they come here forming the mesenchymal cells. Some of these mesenchymal cells, they will displace the hypoblast to yolk sac endoderm, and here they will differentiate into the definitive endoderm. Some more epiblasts will invaginate inward and they will form the mesenchymal cells here that will form the definitive mesoderm. And the cells that remain behind, they will form the ectoderm. So ultimately, through this process of gastrulation, we have three germ layers: the ectoderm, the mesoderm, and the endoderm. So that was about gastrulation. Thank you so much for watching this video.